to introduce to Tamara Seredenina. She is a group leader at the AC Immune, a Swiss biotech company leading research in neurodegenerative disorders. With over 15 years of experience validating drug targets for conditions like amyotrophic lateral sclerosis or frontotemporal dementia, she has made significant contributions to the field. Before joining AC Immune in 2016, she conducted her postdoctoral research not very far from us at the University of Geneva, or EPFL, in Switzerland. Tamara's dedication to the understanding of neurodegenerative disorders is truly admirable, so please join me in welcoming her to the stage. Thanks.
This works now. Yes, perfect. Thank you for the very nice introduction. It's really a great pleasure to be able to speak here today and to share a few thoughts about my career. So thank also for the organizers for the kind invitation. There you are. So let me tell you first about my career path. So I come from Moscow. I did my studies in Russian State Medical University, and that was the first important moment in the choice of a career, because I was thinking whether I want to do medicine or I want to do biotechnology research. And what I really wanted to do is to bring new therapeutics to the patients, and this is why I decided to do the biomedical research. And another important point for this decision for me was also I knew that biomedical research would allow me to work pretty much anywhere in the world, which was very important for me as well. I did my master's degree in biochemistry, and my main interest was always how the brain works and how to improve the brain function. So I worked on neuroprotection, developing neuropeptides that would improve brain function. And after that, I had a fantastic opportunity to move to Italy, to a company called SienaBiotech, where I could continue my PhD. The main focus of this company was Alzheimer's disease, and I was employed in developing new assays for screening and for evaluation of new drug targets in Alzheimer's disease. So working in industry environment gave me an excellent understanding of the needs of a biotech company and also introduced me to multiple technologies. So that was very rich experience. And then in 2008, I came to another pivot point in my career, and I'm really grateful to my supervisor back then, Professor Gerd Verstappen, who told me, what do you want to do next? Do you want to dive deep into technical expertise, or do you want to become a scientific leader? And I decided that to become a scientific leader, I need to go back to academia. And this is how I arrived here to the EPFL. And again, we had a fantastic collaboration between SienaBiotech and Professor Ruth Luthi-Carter at the EPFL in Brain Mind Institute, which allowed me to join her lab and to stay at the EPFL for 3 years as a postdoc, where I was working on Huntington's disease. And the main interest was pathway analysis and understanding of the dysfunction across different neurodegenerative conditions. So this gave me a broad understanding of how brain dysfunctions. And again, I did a lot of exploration of new targets and a lot of validation in models in vitro. And then, as a next step, I wanted also to understand how to move the concept into the next phase, which requires in vivo proof of concept. So I wanted to get more understanding of in vivo research, and this is why I moved then to the University of Geneva in a maître assistant position, where I was working a lot in another neurodegenerative disease, which is amyotrophic lateral sclerosis. And working in this position allowed me to do 3 important things. First is to have a deep dive into neuroinflammation, which is a relatively new concept, which then allowed me to move into my next position. The second thing was to work in a very broad environment with many different partners and to understand how to coordinate the projects and how to coordinate collaborations across different countries, different institutions. And then the third point was a lot of pharmacology work, repurposing actually existing drugs, and that made me realize that what I really want to do in my career is drug discovery. And this is why then I moved back to industry, and I joined AC Immune, a company that's here on campus. So I joined in 2016 as a senior scientist with a small group of 2 technicians, and then over the 7 years, the group has grown into a 15-people group working across 6 different projects into many neurodegenerative disorders, and the main goal of my group is validation of new drug targets. So as you can see, the career path, there is a main red line through my career path, which is based on my main interests, which is in identification and validation of new drug targets and developing therapeutics and diagnostic tools for neurodegenerative disorders. So let me tell you a few words about AC Immune. AC Immune is a company that's pioneering precision medicine. We're based here on the Parc Scientifique campus. The company is more than 150 people now, and it was founded more than 20 years ago. And it had a very successful IPO, so it was listed on NASDAQ since 2016. And the main goal of the company is to develop therapeutics to remove misfolded proteins that accumulate in aging brain, such as amyloid beta plaques or tau tangles in Alzheimer's brain, and Lewy bodies in Parkinson's disease brain. And there are few assets that are in clinical development, so if you are interested, I welcome you to visit our website and explore our clinical pipeline. But for today, I wanted to focus on what my group is responsible for, so it's our preclinical pipeline with novel targets. And you can see that we're exploring 2 areas of novel targets. One is TDP-43 biology, and the second one is neuroinflammation, with a particular interest in targeting NLRP3 inflammasome. And there are 4 different projects that are inside this pipeline. You can see that there are 2 projects developing monoclonal antibodies against TDP-43 and against ASC, which is a component of the inflammasome. And then there are 2 small molecule projects. One is a diagnostic PET tracer program, and the second one is a therapeutic molecule developing inhibitors of NLRP3. And the reason I'm showing you this is to explain you why I really enjoy working in this environment. So my team is responsible for the biology part, so meaning identifying new targets, making screening assays, making validation assays, testing efficacy in vitro and in vivo, and also building the biomarker that would then go together with the therapeutic tool. And all of this is done in a very intense collaboration with our antibody engineering team with regards to the monoclonals, and also with the medicinal chemistry team when we talk about small molecules. So there is a lot of exchange of information and really the synchronization of different parts of expertise. And then, of course, when the drug moves into preclinical pipeline, there's a lot of interactions with pharmacology team, with toxicology team, manufacturing, and then when we move closer to the clinics, we talk a lot to regulatory team and the clinical team. Of course, intellectual property is always involved in these discussions, and there's a lot of project management. And the reason I love it is that we really have the exposure to all of these functions. So being in the medium-sized biotech allows you to get really a full picture of what's happening to your product and how it moves from one phase to another. And I wanted just to give you one example of what we have obtained, what we have achieved with one of our targets, which is TDP-43 as a drug target for neurodegenerative diseases. TDP-43 is a DNA-RNA binding protein that resides in the nucleus, in normal cells, in all the cells in our body. And then in neurodegenerative conditions, it mislocalizes in the cytoplasm and forms this very dense aggregates that then disrupt the normal function of neurons. So what we want to do is to prevent this from happening and to protect the healthy neurons. And this pathology was discovered in 2006 in amyotrophic lateral sclerosis and frontotemporal dementia, but later it was also discovered that TDP-43 accumulates also in Alzheimer's disease and in Parkinson's disease. So there's a very broad therapeutic and diagnostic value of this protein. So what we achieved so far, we have developed a first-in-class monoclonal antibody that targets TDP-43. We've shown efficacy of this antibody in 2 different animal models of amyotrophic lateral sclerosis. We've shown that we can decrease this neuropathology and confer neuroprotection. So this work was published about a year ago. And I'm also showing you this to show that we do publish. We continue sharing our information with the scientific community. And so this product is now in IND phase, and we will move it into the clinical testing. And to ensure the successful clinical testing, we need efficient biomarkers, which are not yet available for TDP-43. So we also have to explore and to propose ideas for new biomarkers. And this is how we started working on a PET tracer, so positron emission tomography tracer, which will allow to image TDP-43 in the brain of living patients. And we discovered a molecule, which you can see here. So these are the images of autoradiography from human brain on control patients and on patients which have FTLD-TDP pathology. And you can see a very specific binding in disease condition, but not in the control. And these images of the monkey brain here show the PET pharmacokinetic profile, where we want to make sure that the compound goes in the brain. So you see a strong signal in the first moment of uptake in the brain, and there is a relatively good washout. So we need to combine the specific binding properties of a molecule with the appropriate pharmacokinetic profile. And this product will be entering clinical testing later this year. And to finish, I wanted to reflect on this question, which you might be asking. I'm getting this question really a lot from students, industry or academia. And to answer to this question, I believe you need to understand a few things for yourself. First is, what would you like to achieve? Where would you like to go in your career? What is important to you? Do you want to make new discoveries and break the boundaries of knowledge? Do you want to develop new drugs? Do you want maybe to travel a lot? Do you want to do more business development? Do you want maybe to have more flexible working time? So consider all of these aspects that are important, especially in our changing working world. And it's not easy to make these choices, so it's really useful to find a role model as someone who inspires you, and also a mentor, so someone who would bring you and accompany you from one point to another and help you to take these decisions. And it's also very important to know what you don't want to be and how you don't want to work, to consider into this question. And once you've sorted this out, the second point is, what is the best environment to achieve your goals? What is the best place that has the right tools and the right skill sets for what you want to do? And in my case, the solution was easy. I wanted to do drug discovery, and I realized that I need to go into biotech industry and pharma industry, which has the most appropriate knowledge and also the resources that are necessary for developing a drug. So that was an important decision point. And of course, it's more complicated than this, and don't be afraid to change and don't be afraid to explore, especially in early stages of career, because there are really multiple opportunities. As we already heard this morning in the first half an hour, we could have exposure to many different roles. And here I've listed just roles that came on top of my mind from the people with whom I did my postdoc, my peers. So some of them became professors, some of them went into teaching, some of them, like myself, doing industrial research, clinical research, business development, intellectual property, medical writing, editorial business. So there are many options, quality, regulatory affairs, we will hear later today. So there are really many options to explore. So what is the right path? What is the good direction? I think it's the one that you take for yourself. And I really like this thought here. So no matter what path you choose, really walk it, which means do your best and bring your positive attitude, because ultimately what people are valued for in the working environment is much more for the attitude and to providing solutions rather than for specific expertise. And also follow your primary motivations. So what drives you? What makes you get up in the morning? This is really important because we put so much energy in our career path, so it's important, and it's much easier to work when you're self-motivated, when what you're doing is really close to your core values. So think about this, and with this I will close, and I would like to wish you a very successful career full of achievements and joy. So enjoy what you're doing. Thank you.